Okay guys, let's talk about smoking and nicotine dependency. According to the CDC, 14% of adults in the United States smoke cigarettes, making smoking the leading cause of preventable disease and death, which accounts for almost 1 in 5 deaths every year. But what makes smoking so widespread? The obvious answer is nicotine, an addictive chemical additive in cigarettes included to keep consumers coming back to buy more. But what makes certain people so susceptible to nicotine addiction? Certain genetic traits change how individuals' bodies react to and handle nicotine intake, which can allow nicotine to have a firmer grasp on an individual, making it harder for them to quit. According to twin studies, 60% of nicotine metabolism is due to genetic factors. It is still being determined exactly how many genes affect chemical dependence, but there are quite a few that seem to affect specifically how nicotine is metabolized in various ways. For example, the gene CYP2A6 and its various alleles affect how quickly nicotine is broken down in the liver, and as such it is produced in liver cells. How fast an individual breaks down nicotine can determine if they get a larger effect from fewer cigarettes, or if they need to consume more cigarettes to attain the same effects as others do with fewer cigarettes. Another gene that has demonstrated effects on nicotine dependence is the chRNA4 gene. The chRNA4 gene provides instructions for making a subunit of a larger protein called a neuronal nicotinic acetylcholine receptor, or NACHR. Variations in this gene can change the rate at which nicotine is taken into the brain, resulting in potentially quicker pleasure reactions in the brain. Although both of these genes affect the body in two different places, it makes sense that they both would have a significant effect on an individual's tendency toward nicotine dependence, as they both affect how the body reacts to nicotine. Let's move on to the SNP chip test. A chromosome single nucleotide polymorphism array is a genetic test that is able to detect changes in a person's chromosomes. If we were able to include variations of every SNP that is somewhat related to nicotine dependence, we would likely have to include 100,000 plus variations. Instead, our group opted to measure 40 variations stemming from 15 different single nucleotide polymorphisms. The two that we found were the best indicators of an individual's susceptibility to nicotine addiction were RS1801272, a single nucleotide variation that affects which type of CYP2A6 protein produced in the liver, and RS1044396, a single nucleotide variation that affects the chRNA4. The chRNA4 gene codes for one of several different proteins that are needed to construct nicotinic acetylcholine receptors. These are ion-conducting channels across the cell membrane. The RS1044296 SNP changes the protein that chRNA4 creates and allows for a faster transfer of nicotine and its subsequent components to affect the individual at a faster rate, thus allowing for a better buzz, but also a need to smoke or ingest more nicotine to keep that buzz. Moving on to the benefits of the test. Due to the genetic nature of nicotine dependence, if one wants to partake in certain activities that involve nicotine, but do not want to do so on a regular basis, then a genetic test will be beneficial. This test will allow for you to understand what SNPs you may or may not have, and will allow for a better decision of whether to take or ingest nicotine. The test will be able to identify all SNP variations on the CYP2A6 gene, allowing for a fuller picture of your likelihood to develop nicotine dependence, as different variations give you a specific percentage of increased likelihood to develop a dependence. This can also help if you decide to quit, as the way these SNPs make you addicted respond better or worse to different nicotine mitigation methods. While there are benefits of the test, there are also limitations of the test. It would be completely irresponsible of us not to inform anybody that was considering taking our test of its potential limitations. Although the mutations that our test is designed to test for are indicators that an individual would be increasingly susceptible to nicotine dependence, there are also many other SNPs that could affect an individual's propensity for substance dependence. It is also likely that there are more unknown SNPs or variations that were unable to test for until further research proves a direct link to nicotine dependence. The mutations that we are aiming to test for cannot measure psychological factors, which may play a role in whether or not an individual uses or continues to use nicotine. But most importantly, regardless of the outcome of the test, nicotine dependence itself is not begun solely by genetic factors. The decision to smoke is ultimately that of the individual in question, and whether or not the individual has certain mutations that we test for does not cause an individual to decide one way or another. The SNPs tested for here are only known to affect how likely an individual is to to have a nicotine dependence after they've already begun smoking. Because of this, an individual that does not smoke may not want to receive this test due to its cost, or the mental effects of being aware of their potential propensity for nicotine addiction. It is possible that knowing whether or not you are more likely to develop nicotine dependence can cause a significant amount of stress, so it is not recommended to take the test if an individual does not plan to begin smoking.